Section 3 of Chapter 12 covers linked genes and how they tend to be inherited together because they are located near each other on the same chromosome. Linked genes are genes that are located near each other on the same chromosome, and they tend to be inherited together. So genes for two different traits. So when we look at, and this somewhat takes a little bit of practice in analyzing the diagrams, writing out a key to kind of keep your brain around what the letters represent. So looking at the first circle in the center, this female she's heterozygous for color. I think this is for a fruit fly. So I see a B plus and a B. So that means she's heterozygous for her body color, and she would have a gray body. Now, the VG together is a different gene, and this is going to be the placement of her wings. VG with a plus sign or a positive sign indicates a normal body placement, and the VG on its own is vestigial wing placement. So this female in the center of the screen is heterozygous for both of those different traits. Now, when she makes egg cells, and these egg cells are going to make little baby fruit flies, we can see that the entire chromosome with the entire genes, and they're pretty close together on this chromosome, they get donated together. So looking at the bottom, the B with the positive sign and the VG with the positive sign, they were on the same chromosome. They got donated to the cell or egg or offspring on the bottom, in the center, or on the left. Looking at the other option in the far right corner, her other letter or allele or gene, B, got donated along with VG together, and they stayed together. There wasn't crossing over taking place where these two different genes got shuffled during prophase of meiosis, early on in meiosis. So these genes, they migrated together. Now, relating these offspring to the traits, the mom was heterozygous, but again, she was having, or is having, a gray body and a normal wing placement. Her two offspring cells down at the bottom don't necessarily have her traits that she has. So in the bottom center, and it also depends on what dad brings to the table as well with those blue chromosomes and his linked genes. But in this example, down at the bottom, on the bottom left, this offspring would also be gray with normal wing placement. And the plus sign indicates wild type, which is normal. And then on the bottom right, this offspring is going to have a black body with vestigial wing placement, which is unique and different than what we saw with the mom. So this can be very confusing. I know coming up there's going to be a few more examples, but I would recommend that you, one, do the reading guide questions. Always write out genotype and how it relates to the phenotype, because it does take a little bit of practice in what all these different letters mean and how it relates to genes being passed on, different letters being passed on, or alleles at the same time. Genetic recombination is the production of offspring with combinations of traits different from either parent. If the offspring looks like the parents, they will be known as parental types, having the same phenotype. If the phenotype is different from the parents, they're known as recombinants. So here's an example. This is a Punnett square, even though it looks different, just to give you an example of what parental type and recombinants are. So we are crossing a yellow round dihybrid parent across the top, crossing it with a homozygous recessive parent that is green and wrinkled. So the two different phenotypes we're looking at, green with wrinkled and yellow with round. So when we do a Punnett square and we look at all the different possibilities of offspring, we have different kinds of offspring. There are yellow and round. There are green and wrinkled. Those would be known as the parental type offspring. And then we got these new ones in the mix. We have yellow, but it's wrinkled. And then we have green, but it's smooth. Those two different phenotypes are known as the recombinants. Yes, they received their gene, their yellow, from one of the parents. Yes, they received the wrinkledness from the other parent, but put together, they don't exactly look like any one parent. So they're known as recombinants. Calculating recombination frequency sounds like it might be a little bit difficult. Figuring it out, all it is a percentage. You can leave it as a decimal or you multiply it times 100 to get a percent. You take the number of offspring that are the recombinants and you divide it by the grand total of offspring. So it's just how many recombinants are there out of the grand total of offspring. And I'll do an example on the next slide. Here's an example that I just went over with the two different P types and what a parental versus recombinant is. We can calculate the recombination frequency once we know the number of offspring that are produced of each different phenotype. So down at the bottom, you can see there were 244 yellow round, 256 green wrinkled, 251 yellow wrinkled, and 249 green round. All you have to do is, as your numerator, add up the recombinants. 251 plus 249 equal 500 of those P's are recombinants. And then you add up the grand total, you add up all those values. There are 1,000 offspring that are produced. So 500 divided by 1,000 is 50% once you multiply it times 100. So 50% of these offspring are recombinants, or 0.50 is the recombination frequency. Here's another sample problem for you to calculate the recombination frequency. Whenever the recombination frequency is determined, the original parents are like true breeding. They only have the same alleles in their genotype. So the P generation or original parents are homozygous. You had one parent with the gray body and normal wings, and then you crossed it with a homozygous recessive parent for black body with the vestigial wings. And that means that all of the offspring are going to be dihybrids or like heterozygous. Then you do a dihybrid test cross. You take a offspring that is heterozygous or like a carrier or a dihybrid, and you cross it with the homozygous recessive partner, basically, and you see what the results are. How many of the offspring are like the parents and how many of the offspring are recombinants. So the numbers are given down at the bottom. And again, in this problem, you would take 206 plus 185 and divide it by the grand total of all the offspring in order to figure out the recombinant frequency. I calculated it to be 83% are like the parents and 17% are like the recombinant. So please double check my math and make sure that you understand how to do it. And I would also jot down the genotypes for what does it mean to have a gray body with normal wings, black body, vestigial, homozygous, and being a dihybrid. So there's quite a bit of information on this slide that I would study and really try to wrap my brain around. If the results do not follow Mendel's law of independent assortment, then the genes are probably linked. So if you have parental values that are higher than 50%, most likely that means that the two genes are linked to each other. When synapsis occurs during prophase and the homologous chromosomes line up next to each other and exchange bits of DNA or bits of the chromosome, it's called crossing over. And this explains why some linked genes get separated during meiosis. The further apart two genes are on the same chromosome, the higher the probability of crossing over that it will occur, and the higher the recombination frequency should be. But as I previously stated, if the frequency of parents is higher, this means that crossing over most likely did not occur and the two genes are staying together. A linkage map is a genetic map that is based on the percentage of crossover events, and it's based on the recombination frequency. So you might see something as map units. So basically what you do is you look at your recombination frequencies of the different genes that you have, and you try to place them in order of being really close together or really far apart. And you're going to be given, or you'll have to calculate, the actual recombination frequencies. It expresses the relative distance along the chromosome, and 50% recombination values means that they are very far apart on the same chromosome, or possibly they could be on two different chromosomes. And what you're looking at in the picture is a chromosome, that's what the purple is, and you're being given the values between three different genes. B versus the VG, they're very far apart, so they have a recombination or a map unit of 17 or 17%. And then CN is basically directly in the middle, 9% away from the B gene and 9.5% away from the VG. So you'll be given values, and it's a little bit of a mystery, on how to place these in order on the chromosome. The past couple of chapters, we've really focused on Mendel and how genes are passed on to offspring and ultimately expressed in the offspring's phenotypes. But there are exceptions to Mendelian inheritance. An exception to Mendelian inheritance is known as genomic imprinting. Genomic imprinting is where there's a phenotypic effect of a gene depending on whether or not this gene came from mom or dad, or from the male or female parent. There's a diagram or an example down at the bottom, a normal-sized mom mouse versus a smaller dwarf dad mouse. Looking at the mom, she has two genes. A normal allele is expressed, and the mutant gene is not expressed. And with dad, the mutant allele is being expressed, and that's why he has the size that he is, and the normal gene is not being expressed, even though both of these mice are heterozygous for both these genes for size, basically. Another example of an exception to Mendelian inheritance, is called methylation. Now, this is different than the example that's being shown, but methylation can silence different genes by simply adding methyl groups, which is the CH3, if we remember that from a long time ago, to DNA. The more methyl groups that are added to DNA, the more likely the gene will not be read. Something we shouldn't forget when we're studying inheritance is that there are other locations of DNA, non-nuclear DNA that's not located in the nucleus, which is primarily everything that we've been talking about up to this point. Some genes are located in organelles. What are these organelles? Mitochondria, the chloroplasts, other plastids. They contain small circular DNA. And mitochondria has maternal inheritance. There is DNA passed on into the egg cells. An example using plants, striped or spotted leaves in the picture that's being shown result from mutations in the pigment greens in these plastids, which are generally inherited from the maternal parent. Do you want to say hi?
Hi everybody, it's me Max. My mom is talking. So I got a new toy for Christmas and some jazz for my mom and dad. Bye! People, my name is Will. I wish you a Merry Christmas and a Happy New Year and a Merry good night.
How about
See you later.
Oh, okay.
Bye bye.